More than 50% of the dry mass of cells is protein, making protein a very important structural and functional part of living cells. In this video, we will have a closer look at the structure of protein molecules. Proteins have many important functions. Proteins are essential components of cell membranes. Hemoglobin is an oxygen-carrying protein and pigment. Invading microorganisms are attacked and destroyed by antibodies, which are proteins. All enzymes are proteins. The strength of many tissues, like bones and artery walls, depend on collagen, which is another protein. The surface layer of our skin and hair contains keratin, which is a protein. Proteins are macromolecules and polymers, meaning they are large molecules built from small building blocks or monomers. The monomers of proteins are amino acids. Amino acids have a central carbon atom and a carboxylic group and an amine group on either side of the carbon. It is these two groups which give the amino acids their name. The third component bonded to the carbon atom is a hydrogen atom. The fourth group bonded to the central carbon is the R group, which varies on different amino acids and which is the only group on the amino acid that differs. There are many different R groups, which can produce 20 different amino acids that occur naturally in proteins of living organisms. Many other amino acids have been synthesized in a lab. It is these R groups which are responsible for the three-dimensional shape of the protein molecules and therefore its functions. Amino acids are bonded together by a bond called a peptide bond. One amino acid will lose a hydrogen atom from its amine group, and the other amino acid will lose a hydroxyl group from its carboxylic group. The first amino acid's carbon atom is then free to bond with the next amino acid's nitrogen atom with a bond called a peptide bond. The oxygen and the hydrogen atoms removed can form a water molecule during a reaction called condensation. This type of reaction also happens when glycosidic bonds in carbohydrates form and when an ester bond in a triglyceride forms. Two amino acids linked together is a dipeptide. More amino acids can be added to the chain during condensation reactions. Many amino acids that are linked together by peptide bonds are called a polypeptide. A polypeptide is another example of a polymer or a macromolecule. A complete protein molecule may contain one polypeptide chain or could have two or more chains which interact with each other. Ribosomes are the site where amino acids are linked together to form a polypeptide in living cells. This reaction is controlled by enzymes during the translation step of protein synthesis. If we want to break down the polypeptide to amino acids, we need to break the peptide bonds between the individual amino acids. This involves a hydrolysis reaction with the addition of water, and it happens naturally inside the stomach and small intestine during digestion. Protein molecules in food can be broken down or hydrolyzed to amino acids just before they are absorbed into the bloodstream. There are 4 levels of structure in proteins. The first 3 levels involve a single polypeptide chain, and the 4th level involves a protein that has 2 or more polypeptide chains. Primary structure. The primary structure of protein is when 3 or more amino acids are linked in a straight-chain polypeptide molecule. The type and sequence of amino acids are determined by the code in DNA. Polypeptides can contain several hundred amino acids that are linked together in a straight-chain molecule. The types of amino acids and sequence of amino acids determine what type of polypeptide is formed. There exist several different possibilities of combinations of amino acids, literally thousands. The primary structure will change even if only one amino acid in the chain is left out, added, or swapped with another. This small change may change the properties and functions of the polypeptide. The properties and function of the polypeptide or protein are thus determined by the DNA code. Secondary structure. The secondary structure of a protein occurs when the weak negatively charged nitrogen and oxygen atoms interact with the weak positively charged hydrogen atoms to form hydrogen bonds. Most fibrous proteins have secondary structures, for example, collagen and keratin. The secondary structure only relates to hydrogen bonds forming between the amino group and the carboxyl group, the protein backbone. The amino acids in the polypeptide chain influence each other even if they are not directly adjacent or next to a specific amino acid. There are two types of secondary structure, and both types are held together by hydrogen bonds. A polypeptide will coil into an alpha helix due to the attraction of the weak, negatively charged oxygen atoms of the carboxyl group of one amino acid and the weak, positively charged hydrogen atoms of the amine group of another amino acid 4 places ahead of it. A hydrogen bond forms between these groups due to the polar nature of the oxygen and hydrogen atoms in these amino acids. The hydrogen bonds can keep the alpha helix in its shape, but can be broken by pH changes and high temperatures. These changes in the environment can have serious consequences on the proteins of living organisms. Some proteins form a much looser, straighter shape called a beta-pleated sheet and do not form an alpha helix. The polypeptide can fold so that 2 parts of the chain are parallel to each other. Then the beta-pleated sheet shape forms, which enables hydrogen bonds to form between parallel peptide bonds. Some proteins show no regular arrangement. It depends on which groups are present and therefore what attractions there will be between the amino acids in the chains. In many proteins, the secondary structure itself is folded or coiled. A further conformational change occurs, and more bonds are added in the protein structure. At first sight, such a folded molecule looks like a disorganized tangle, but it is in fact highly organized. The shape of the molecule is very precise and held in exact shape by bonds between amino acids in different parts of the chain. It is determined by the way in which the protein coils to form a precise 3-dimensional shape known as its tertiary structure. 4 different types of bonds can form between amino acid R groups. Hydrogen bonds can form between a wide variety of R groups, including those of tryptophan, arginine, and asparagine. Disulfide bonds form between 2 cysteine molecules. Ionic bonds form between R groups containing amine and carboxyl groups. Hydrophobic interactions occur between R groups that are nonpolar or hydrophobic. Tertiary structure conformations are common in globular proteins like antibodies. Quaternary structure. Many proteins are made of 2 or more polypeptide chains which interact with another polypeptide chain. Each chain is called a subunit and is held together by 4 types of bonds which are also seen in the tertiary structure. The quaternary structure of protein may have prosthetic groups, which are non-protein groups associated with the protein. These prosthetic groups give the protein its specific function. The distinguishing factor between tertiary and quaternary structure of protein is that there is more than one polypeptide chain in the quaternary structure. Fibrous and globular proteins can have quaternary structure. Tertiary structure proteins also do not contain prosthetic groups. I hope that you have a clearer understanding of the basic structure of proteins. Let me know in the comments what else you would like to learn about.